Howdy, folks. In this video, we are going to talk about bacterial transformation. So with bacterial transformation, what we're going to do is change the DNA of a bacteria. And we're going to do that by inserting what is known as a plasmid. So a plasmid is just a small amount of DNA, usually from the environment. And in order to get the bacterial to change its DNA, there are several steps involved. The first thing that we would do is use what is known as a restriction enzyme to break open the section of DNA that we're looking for, and that is done with just using enzymes that are commercially available. So with this, what we're going to do is take that specifically sectioned off gene, that section that we are interested in, and then we're going to insert it into the bacterial DNA. So we're going to cut the bacterial DNA. Remember, bacterial DNA is circular. We're going to take that gene and we're going to join them together and glue them together, a process known as ligation. Um, After the ligation piece, then we're going to do the actual transformation, and hopefully it works. Now, with the transformation process itself, this is the part that I'm going to go into a little bit more details about. So we have to get the DNA into the bacteria. Then we have to check to see if the bacteria actually took in that DNA. So we're going to talk about the transformation process itself, using this simulation diagram to kind of help you understand. Now, this original video does go into more information that you need to know, so that's why I'm recording over it. First of all, we have the plasmid itself, and we're going to need to insert it into the bacterial DNA or the bacterial itself. But keep in mind that this prokaryotic cell, this bacteria, is surrounded by a cell membrane. And so that cell membrane is semi-permeable. We talked about this back in Unit 2. And with that semi-permeability, it's going to prevent DNA from coming in. DNA would normally be uh, rejected or not allowed through because DNA has a charge. DNA has a negative charge. And so those hydrophobic tails are going to prevent it from passing through. The process of transformation will then take that membrane and break it up, actually make breaks in the membrane, and that's going to allow the DNA, the plasmid that we want, to enter into the cell. Now, when we are looking at this transformation process, there's a chance we could accidentally kill the bacteria as well. So we're going to have to check to make sure that the bacteria actually survives the transformation process, because we are going to do what's known as the heat shock. So the first thing we're going to do is we're going to take that plasmid and we are going to digest it, and we are going to input the cells that we want. We kind of take this mixture of the digested enzyme, the digested plasmid digested by the enzyme, the cells, and we cool it down. And in this process, you do have to let it cool for a while. We're trying to get it to change temperature so that we can do what's known as heat shock. Now, heat shock sounds just like what it sounds like. We're going to submerge it in a warmer bath. And with this, it's going to create that shock that's going to break up the plasma membrane itself. So the heat shock happens very quickly. And then we're going to cool it back down so that the membrane can essentially heal itself. And hopefully in this process, the bacteria took in, because the membrane was opened up, hopefully in this process, the bacteria took in the plasmid, the inserted gene that we wanted it to have. Now, this is something that happens to bacterial cells. You can see from this process, it's not going to be as easy. This is definitely something that would happen in a lab setting, but our ultimate goal of this is to change the bacterial DNA, because we can use this then to produce useful proteins. Remember that DNA codes for proteins. And so you can use this to code, for example, human insulin. You can transform the bacteria so now they become insulin making factories because insulin is just a protein. And so now you can get these bacteria to produce desired proteins. So once the bacteria has taken in and hopefully taken in the plasmid, we are going to then incubate it. We're going to take some samples and we're going to grow it in some media. Now, you don't need to know the details about this media, but what you do need to know is that we are going to start growing the bacteria to see if it was successful. So this is how we're going to do that part. So what you're seeing on here is the bacterial transformations, the results of this bacterial transformation. So this is how we would test it. Before we even start this bacterial transformation process, we're going to have, these are plates where we grow the bacteria on, and on them is agar. So agar is a substrate that the bacteria will grow on, and it's going to have nutrients. So what's going to happen is before the transformation process, we're going to check to make sure that this bacteria actually grows. It's actually alive. And we can do that by growing it on this agar. And so what you see here are many, many colonies. These circles represent colonies, millions of bacteria. And you can see that they're all kind of spreading. So we're going to confirm that the bacteria actually grew on our plates. Then we're going to go in and check to make sure that whatever it is that the bacteria is doing, we're going to have a way to check to make sure before and after, did it change? So in this example, we're going to be looking at antibiotic resistance. So this plate is a different color because the agar also has an antibiotic. Now, if this is normal bacteria, then no colonies should appear on this antibiotic plate. That's what we call antibiotic sensitive. So on here, you see no colonies of bacteria. That's how we know that this bacteria was sensitive to the antibiotic. But what we're going to do in this transformation is we're going to try to see if we can make the bacteria resistant to the antibiotic. So then we perform the transformation, the heat shock, as mentioned, the cold, the hot, the cold, all of that stuff, the digestion of the bacterial DNA, the plasmid. And then what we're going to do is we're going to go back and test to make sure that the bacteria actually survived. Remember, I told you it's a heat shock. We're opening up that plasma membrane. There's a chance that the bacteria could die in that process. So we're going to check to make sure that did it die. And you can see from this plate, there is growth. So we have confirmed that the bacterial did survive after the transformation. Now, on this last plate, what we're going to do is check to see, did the bacteria take up the plasmid? And this plasmid, or the gene, was for antibiotic resistance. So this plate has antibiotics. That's why it's the same color as this one. And what you'll see is that you do have some colonies on this plate. Now, the reason why you have colonies is because now the bacteria has taken up the plasmid and is resistant to the antibiotic. But not all the bacteria survive. So you have less bacteria that survived, not only survived the process, but actually took in the gene. So you are going to see some growth, but not all the bacteria took in the plasmid or incorporated the plasmid. So not all the bacteria is going to be antibiotic resistance. And that's why you see less colonies on this plate here. Thank you so much for watching, folks. Bye.